This protocol can provide specially resolved information about protein conformational dynamics, which can help to identify functionally important regions in protein of interest. This technique probes protein conformational dynamic under near-native condition without the need for protein labeling, using nanomolar amount of material for each reaction.
Begin by preparing undeuteurated reference samples for the protein of interest in triplicate in 500 microliter tubes. Quench the samples with the appropriate volume of quench buffer per tube to achieve a pH meter reading of 2.5. Flash freeze the samples in liquid nitrogen for storage at -80 degrees Celsius until their analysis. Then analyze the proteated enzyme reference samples using a bottom-up liquid chromatography mass spectrometry workflow according to standard protocols. To analyze the raw mass spectrometry data in the appropriate proteomics software program, navigate to Libraries and Protein Sequence Databanks to import the amino acid sequence of the protein of interest. Enter a name for the protein sequence of interest and import the protein sequence in FASTA format. To define the processing parameters, under the Library menu, select Electrospray MSE as the data acquisition type. In the Lock Mass for Charge 2 field, enter 785.8426 for the mass to charge rate.